Human cloning. Cloning is the process of creating an identical copy of an organism or cell. Human cloning is specifically cloning an entire organism's genes to get the exact same organism, in this case being a human. Cloning happens all the time when organisms reproduce asexually in the environment. Unlike human cloning, in nature there is no need for recombination and changes in the genes. There are three main ways humans can be cloned. The three main ways are gene cloning, reproductive cloning, and therapeutic cloning. There are many other techniques that are used for every individual type of cloning, like somatic cell nucleus transfer or gel electrophoresis. Gene cloning produces copies of genes or segments of DNA. Reproductive cloning produces copies of whole animals and was first performed on a salamander embryo in 1902 by Hans Spemann. Therapeutic cloning produces embryonic stem cells for experiments specifically to create tissues to replace injured or diseased tissues. In gene cloning, recombinant DNA is used. So a plasmid is taken and a section of it is replaced with the gene of interest. In this case, everything would need to be replaced as we want the whole human to be cloned and identical. Reproductive cloning techniques underwent significant change in the 1990s following Dolly, who was cloned through somatic cell nucleus transfer, which is when its nucleus is removed and placed into an unfertilized egg that has also had its nucleus removed. She was cloned at the University of Edinburgh, Scotland, by Keith Campbell, Ian Wilmot, and their colleagues. Referring to what we learned in class, in order for a human to be cloned, it does require DNA and RNA. This means in order for the egg to develop to an embryo, then a fetus, DNA replication is used to replicate the DNA that is needed for the egg to develop and become identical. Just as DNA replication is used for DNA for the clone, RNA is needed, so transcription and translation is also needed, as the clone will need RNA to get the required proteins provided through the amino acid produced through transcription and translation. These factors will be further discussed and explained in different techniques that will be shown.